Brown. I'm a science and healthcare reporter here at GenoWeb. I'll be your moderator today. The title of today's webinar is Automating Single-Cell Sequencing Workflows to Generate Cell Atlases, and is sponsored by SP2 Labtech. Our speaker today is Cheryl Paul, Research Associate 2 at Chan Zuckerberg Biohub. We have Ritwik Argel, Research Associate at Chan Zuckerberg Biohub, and Melissa Netwell, a field application scientist at SPT Labtech. You may type in question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of wedges to enhance your webinar experience. With that, I will turn over to Cheryl for her presentation.
Hello, everyone. Hope everyone can hear me. Thank you for coming to our talk today. I'm Cheryl Paul, a research associate from Chan Zuckerberg Biohub San Francisco, and I'm joined by my colleague Ritwik Arjal, and we will be talking about how we have incorporated automation into single-cell workflows to generate cell atlases. A little background on what the Biohub is. The Chan Zuckerberg Biohub Network is a group of nonprofit institutes that brings together scientists, engineers, and physicians with the goal to pursue scientific challenges. We focus on understanding underlying diseases and developing new technologies that will enable actionable diagnostics and effective therapies for diseases. The San Francisco Biohub was in SF was launched in 2016 and focuses on elucidating dynamic cell systems in health and disease. We work in collaboration with our 3 partner universities, Stanford University, University of California, Berkeley, and UCSF. We have 3 major focus areas at the SF Biohub, namely quantitative cell science, infectious diseases, and the technology platforms. Each focus area has multiple groups working on tackling a variety of problems. For example, intracellular architecture, physical biology, human disease, quantitative tissue morphogenesis, virus host interactions, replication, and in the technology platforms, we have genomics, bioengineering, mass spectrometry, computational microscopy, and data science. Over the past 7 years, there have been multiple tools that have been built at the Biohub network, which are all open access and have greatly contributed to the scientific community. Some of them are mentioned here, which is Tabula Sapiens Cell Atlas, Open Cell Atlas, Zebrahub, the Global Outreach Program, CZID, CZ Cell by Gene, so forth and so on. If anyone is interested in any of the tools or wants to take a deeper look into these tools, please feel free to scan the QR code on the top right, which will take you to our website. So moving on to the genomics platform. As part of the genomics platform at CZ Biohub SF, our goal is to continuously evaluate and integrate cutting-edge technologies and methodologies to enable the utilization of high-quality genomic measurements to advance biomedical research. We have 3 primary focus areas, namely sequencing, single-cell sequencing, and spatial transcriptomics. Starting with spatial transcriptomics, it is the latest technology in genomics which not only allows you to measure all gene activity in a tissue sample, but also allows you to map the region where the activity is occurring. We have 3 major technologies for spatial here at SF. The first one is VisGen, which is an imaging-based technology on the left. And then we have 10x Visium and CurioSeeker, which are sequencing-based approaches. The advantages of sequencing-based approaches is that you do not need to know the gene expression in the tissue prior to doing the experiment. Whereas with imaging-based, since you need to build custom probes specific to the gene of interest, you need to know this expression information prior. But VisGen lets you look at the subcellular level compared to the other technologies, which are currently at a multicellular level right now. 2 of the projects that we wanted to highlight that we've used for these technologies for are the mouse lung tuberculosis project and the mouse brain glioblastoma project. At the bottom, left and right, you can see in the mouse brain glioblastoma, Visium section clearly shows the difference between the healthy and the tumor regions of the mouse brain. Moving on to sequencing, we have three types of technologies available for sequencing at the Biohub, namely Illumina, PacBio, and Nanopore. Illumina is short read based sequencing, and PacBio and Nanopore are long read based sequencing. With Illumina, it can give you about 50 to 600 base pairs in size and anywhere between 1 million to 12 billion reads. So it's extremely high throughput. With PacBio and Oxford Nanopore, we can sequence really long fragments of DNA or RNA from anywhere between 10 to 30 kilobases. But the throughput is really low compared to short read sequencing. So depending on the question that you want to ask and the throughput and the size that you need for analysis, you can either go with short read or long read sequencing. Finally, coming to single cell sequencing. So why single cell sequencing in the first place? In bulk sequencing, we get an average gene expression in a population of cells. There's no way to know which expression belongs to which cell type in a tissue, as you can see here in the top. Single cell sequencing solves this problem where you can identify gene expression in each cell type. This provides higher resolution and can be used to discover rare cell populations and provide an insight between different cell types in the same tissue. There are two types of single cell technologies at CZ Biohub SF. One is 10x Genomics, which is droplet-based, and SmartSeq2, which depends on FACS sorting of cells into individual wells of a plate. The throughput of 10x is much more higher, where you can work with thousands of cells at the same time compared to SmartSeq2, which is limited by the number of plates that you can process at a time. One of the examples of a single cell project is the Tabula Sapiens Cell Atlas project, which is a multiple organ single cell transcriptomic atlas of humans. It's a molecular reference atlas for more than 400 cell types of the human body. The Tabula Sapiens Consortium used single-cell transcriptomics to measure the messenger RNA molecules in each of nearly 500,000 cells from 24 tissues and organs. So this was a huge collaboration and a huge initiative, which obviously needed a lot of automation, and which is why we are all here to talk about automation. So Ritvik here is gonna take over and talk about how we've incorporated automation into our workflows.
Thank you, Cheryl. So when considering all the different biological applications and throughput of workflows, variable amounts of preparation will be necessary based on the design of the study. So in larger scale studies or atlas building, there's a need to scale up the sample preparation to make efficient use of the time and your reagents. So if you're doing 1 to 12 samples, then you choose a single or a multi-channel pipette. If you're doing 12 to 48 samples, you would choose a multi-channel pipette and process your samples in a plate format. But if you're doing high throughput workflow where you're processing more than 48 plus samples, then you want to choose a semi-automated or an automated liquid handler to scale up the preparation of your libraries. Additionally, these liquid handlers will make other manually tedious parts of your workflows, such as normalization, pooling, stamping, copying, easier, which helps in scaling up the library preparations. So at CZ Biohub SF, there are a lot of liquid handlers that are available for use to automate high throughput workflows. The Mosquito, Dragonfly, and the SPT Firefly are the more commonly used ones to scale up or automate the RNA-seq or single-cell RNA-seq workflows that were required to generate these atlases at CZ Biohub. So one of the single-cell RNA-seq protocol that was used during Atlas projects was SmartSeq2 protocol. The SmartSeq2 workflow is a plate-based single-cell RNA-seq workflow. The SMART here stands for a switch mechanism at the 5' end of RNA templates. So in this workflow, you have single cells that are lysed in a buffer that contains free DNTPs and DT-tailed oligonucleotides with a universal 5' anchor sequence, after which reverse transcription is performed. After the reverse transcription, you add a TSO, which carries 2 riboguanosines and a modified guanosine. After the first strand reaction, the cDNA is then amplified using a limited number of cycles. After you have the cDNA is then tagmented and further amplified and is used to construct the sequencing libraries quickly and efficiently. So this protocol allows for improved read coverage across the transcripts. And if there's improved coverage across the transcriptome, it allows for better detection of alternative transcript isoforms and SNPs. One good thing about this protocol is you don't need to know the mRNA sequence to follow this workflow, and the reads that you get from this protocol are usually highly mappable. So here at CZ Biohub SF, the SmartSeq2 protocol has been automated using 2 liquid handlers and in 2 stages. The first stage is the preparation of cDNA from RNA in lysis plates. So this part of the protocol needs to be done in a pre-PCR environment. So lysis until cDNA synthesis has been automated using the SPT Dragonfly liquid handler. So one feature of this automated protocol is that the cDNA that is generated here is enough for multiple rounds of library preparations if necessary. It is so because the workflow has been optimized to use lower amounts of cDNA in the downstream preparation of the libraries. The second half of this protocol, which involves tagmenting the produced cDNA, neutralizing the, say, tagmentation reaction, and the PCR, has been automated on the SPT Firefly. So all automated steps for this protocol or for this section of the protocol happens in a 384 well format so that you're getting the most out of the hands-on time that you will have to invest when carrying out this protocol. So what is the benefit of having this protocol automated, right? So one avenue to explore is reagent use. Especially when conducting a large-scale study or Atlas project, all costs will add up, especially if you're processing upwards of, around 100, 150, 384 well plates. The automated protocol that we used is 1/10 volume compared to the regular protocol. So not only do you use liquid handlers to reduce the reagent use by 10-fold, there is also a reduced cost per cell. Now, if you see on the right-hand side of the table, the volumes that are required for the 384 well plates are less than 1 microliter, which tends to be more error-prone when done by human hands. Using liquid handlers such as Firefly and Dragonfly, which are capable of handling low-volume liquids, will help you mitigate that error issue. So not only do you use less reagents, the downstream library prep has also been optimized in a way where you can go with as low as 0.1 nanograms of cDNA to get similar level of information. So now the library prep kit that we use for this workflow is, and that has been automated, is a Nextera XT kit from Illumina. And having that protocol automated was helpful because that kind of library prep where you have to tagment and amplify is useful for a lot of different studies, and it is not just limited to RNA-seq or single-cell RNA-seq workflows. And finally, filling out 384 well plates by hand obviously takes much longer than using these liquid handlers to fill out plates. Okay, so now that we know that we can save on the reagent use, how good does the data look when you prepare samples via automation or by hand? So to compare or to understand this, a small verification study was done here at CZ Biohub SF, where 16 cells were taken and made into 16 cDNA molecules, and from each cDNA, libraries were made either manually or via automation. So the RNA to cDNA was automated on the Dragonfly, and then the quality of the cDNA to the library prep was the one that was questioned. So this slide shows a distribution of the cDNA size and the length, and also the tape station trace overlay for all the 16 samples. The average cDNA yield for this study was 4 nanograms per microliter, and the average cDNA size, as you can see, is around 2150 base pairs. So the size comparison of manual versus Firefly tag-minted libraries is shown in this slide. So it shows that the sizes of the libraries are comparable to one another. However, this is true in a scenario of 16 samples. If the 384 well plate was prepared manually, the sample in the first few wells will be tag-minting for much longer until you're done with wells 383 and 384, which makes a strong argument for choosing automation for size-sensitive library preparations and workflows. I mean, this is going to be valuable, especially when you have to conduct a large study where you have to process multiple samples within a short amount of time. So this slide shows a comparison between library yields between the manual prep and the automated prep. The Firefly library prep is more consistent with a much smaller standard deviation versus the manual, which varies a lot. Again, bear in mind that this is only a comparison with 16 samples, and if someone had to process 384 samples instead of just 16, I would expect the manual standard error to either stay there or go up. So from this, we can say that the Firefly or the automated library prep is more consistent in preparing the libraries, at least in terms of the library yields. So after the libraries were made, all automated samples were pooled together and all the manual samples were pooled together by equal volume. These two pools were then further pooled together equimolar into a single sequencing pool, which was then sequenced. The traces of those libraries are shown in this slide. The manual pool was 30 base pairs shorter than the automated library pool, which was 30 base pairs longer than the manual. So this is what the final pool looked like with the size of around 700 base pairs and a yield of around 9 nanomolars. This was then sequenced on a MiSeq V2 micro to get an average of 110,000 reads per sample. The manual samples did get 1.3x more reads on average, so the differences in the total read count or the genes detected can be accounted to this difference in read depth. So after the samples were sequenced, we first wanted to check whether the libraries pass the single-cell RNA-seq QC metrics. So first thing we wanted to check was whether the amount of mitochondrial DNA that is coming through is the same in both types of sample preps. From the violin plot on the right-hand side, you can say that the amount of mitochondrial DNA that's coming through is about the same. Both total counts for the sample and number of genes by counts are presented in a log scale. The total counts for the samples are in the same order of magnitude between the manual and the automated preparations. Similarly, the number of genes by count metric also shows similar values between the manual and the automated libraries. So to check one step further, we checked the number of genes per cell in this study. Let me preface by saying that the number of reads for the manual preparation was 1.3 times higher than the Firefly reads. The manual preparation did have a higher genes per cell in this study. However, normalizing counts for SmartSeq2 data is still an ongoing field of research, so there was no read depth normalization or downsampling done on this study, even though the manual samples did get more reads than the automated samples. Additionally, there was a Boolean filter for gene detection, so any gene that even had a single count is accounted for in these plots. Having said that, with all these facts considered, the number of genes detected on the manual was more than the number of genes that was detected on the Firefly. So this slide shows a count correlation comparison between and within the methods of library preparation. The correlation within the samples that were prepared manually is similar to the correlation within the samples prepared by the Firefly. Additionally, the overall counts seem to be well correlated between the manual preparation and the Firefly preparation, shown by the heat map on the right. So this tells that the libraries that are prepared by the Firefly are able to capture similar level of complexity as the manual preparations. So the liquid handlers shown here are integrated into our workflows, especially high throughput workflows. The Mosquito is predominantly used for column dispensing or column pooling. The Dragonfly is used for plate dispensing from reservoirs, so this can be used to automate protocols that require dispensing reagents into plates. The Firefly, which is the most versatile of all three of them, is used for all things that the other two are used for, but also used for bead cleanups, library preparations, pooling, copying, and normalizing plates. So the normalization feature, this is one of my favorite features of the Firefly, where it is very easy to normalize a plate as long as you have a corresponding CSV for the liquid volume that needs to be dispensed into the desired wells of that plate. So the UI shows, liquid amounts and helps you visualize by relating color intensity to the dispensing volume. So this makes normalization of large number of samples or plates very quick and less error prone. This allows us to, reliably scale up preparations for larger studies where we are processing and normalizing a lot of plates in the same time. So automation overall enables us to process 384-well plates quicker than by hand. Automating the library preps and sample prep is useful because it's less error prone than processing the samples by hand. Using liquid handlers and automation machines that are able to handle small volumes helps us to miniaturize our reactions, which in turn allows us to save reagents. Considering one of the single cell workflows that was talked about in this presentation, we can say that this particular workflow allowed us to generate more consistent libraries when using automation to generate the libraries. The libraries that were generated using this automated workflow had similar RNA-seq QC metrics, which tells us that the library quality is comparable between automated libraries and the manual libraries. And all the tedious processes, or especially manually tedious processes such as pooling, normalization, copying, and stamping, is automated, which makes it much easier to scale up and process as many samples as we can in a given amount of time. So all these features allow us to scale up the plate processing to enable us to process 6 384-well plates in around 4 hours of library preparation time. So with that, I would like to acknowledge the Chan Zuckerberg Network, the Chan Zuckerberg Biohub San Francisco, the SPT Labtech, and our 3 partner institutions at the Biohub. I would also like to thank my team and my fellow colleague and presenter here, Cheryl Paul, for her help and our team's help in continuous feedback and helping us put this presentation together. So if you want to learn more about Chan Zuckerberg Biohub San Francisco, feel free to scan this QR code, which will take you to our website where you can go through all the tools that have been built here and go through what Chan Zuckerberg Biohub San Francisco is all about. So with that, I will hand it off to Melissa, who's a field application scientist for SPT Labtech.
Hi, everyone. Sorry about the little technical glitch on my end there. Thank you so much, Ritwik and Cheryl. That was a really great presentation. We really appreciate everything that Chan Zuckerberg does in general. The entire team has been really great to work with, so thank you. As Ritwik said, my name is Melissa Netwall. I am one of SPT Labtech's field application scientists. I'm currently based in the Bay Area, but soon will be based in Seattle, covering the entire West Coast. This time was meant for me to introduce the Firefly, but Cheryl and Ritwik have done a really great job for me, so this should be fairly short and sweet on my end. All right, so for those of you who are not familiar with SPT Labtech, I'll take a quick minute to introduce 2 of the liquid handling platform instruments that went into designing the Firefly. First, the Dragonfly is a non-contact dispenser, utilizing true positive displacement with a dynamic volume range. 4 mils, which is the entire capacity of the syringe, down to that 200 nanoliters, that low volume that Ritwik was mentioning earlier. This instrument was released by SPT over 10 years ago. And then second, we've incorporated a air displacement head from Apricot into the Firefly as well. Apricot has been in the liquid handling space for over 20 years now, and they were acquired by SPT in late 2021. So SPT and our brilliant engineers went on to combine these two existing well-established technologies into one compact instrument that was designed for scientists with genomics in mind. Firefly was released at SLAS a couple of years ago, and Chan Zuckerberg was one of our earliest adopters of the system. So moving on to the Firefly itself, this is the Firefly. It has two heads, like I mentioned earlier. It has the Dragonfly-like non-contact dispenser, and then Apricot's dual-core pipetting head. That dual-core pipetting head is capable of picking up 384 well tips, 96 well tips, and then strip tips of 8 or 16 without changing out the core of the head. In addition, it also has 16 deck positions, an integrated gripper, and process modules like a bioshake, a plate thermal module, and a reservoir thermal module with temperature ranges from -10 up to 99°C, enabling users to keep reagents and samples at temperature, all in a compact instrument that can fit on a standard benchtop. And this instrument is a really great fit for experienced automation users, but also those who are looking to get started in the automation space. If you happen to be in the latter pool of people who are attending this webinar, we did another webinar with TGen's Genomics and Pathogen Division earlier this year, where they discussed the decision-making process around moving from manual to automated library prep for their projects. I also just want to take a minute to call out the reagent savings enabled by utilizing the dispense head for aliquoting your reagents. So a lot of times when you move to an automated system, you will have to utilize the SBS footprint reservoirs. A lot of times those require on the order of mils of dead volume. So by utilizing the dispense head, you're able to scale that down pretty significantly. And so we currently have two types of reservoirs for both the Firefly and the Dragonfly. The standard has a 10 mil capacity with a 240 microliter dead volume, 200 of that being fully recoverable. And then we have a low dead volume reservoir, which has a 1.5 mil capacity with a 75 microliter dead volume, with 35 of that being completely recoverable. And to wrap up, the Firefly is chemistry agnostic platform. We've developed and optimized methods for multiple library prep kits, which can be found on our community cloud and are free to download by any of our users. We don't charge per script. And like I said earlier, the instrument was designed for scientists. The UI is incredibly intuitive and flexible, while still allowing for a great deal of control over the liquid handling parameters. And finally, a note on the support team. SPT has an incredibly engaged team of application scientists. After installation of this instrument, one of us will spend time with you to help develop and optimize any methods that we have not already published to our community cloud, so that you're up and running by the time that we've completed applications training, which has been really, really nice for our current customers. And please feel free to reach out directly or place an inquiry through our website if you have any questions or want to learn more. Thank you again to the Chan Zuckerberg Biohub for sharing their work and experience using the Firefly, and thank you to everyone for joining. And with that, I'll pass it back to Hunja.
Thank you, Melissa. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We would like to ask attendees to take a moment after the webinar has ended to take our exit survey to give us your feedback. We'll now start the Q&A portion of the webinar. The first question is, how much time taken to complete the first round of Tabula Sapiens cell atlas?
Hello. It took us about 3 years to complete the whole first round to the entire cell atlas, but keep in mind it's a human cell atlas, so we spent a lot of time waiting around for donors for tissues. So a lot of time was spent to wait for the donor tissues, but overall it took us about 3 years.
Great. there's a question from the audience saying, how is the yield of the reverse transcription in your hand?
Hello.
The question is,
sorry. Yeah. So when you mean reverse transcription, do you just mean before the cDNA amplification or after?
Unclear. I think the question is worded the yield of reverse transcription. Maybe the audience can.
I see.
The question. Yeah.
Because I haven't. I don't really QC right after there's the reverse transcription reaction without doing the cDNA amplification. The numbers for the cDNA amplification are on this slide here, where the average yield after the reverse transcription and the cDNA amp is around 3.96 nanograms per microliter. And usually you get around 15 microliters, so around 60 nanograms total.
Yeah. Next question. So we're working on circulating tumor cells and isolate those cells in numbers from 1 to 400. So which method would you recommend? Any takers?
Yeah.
That's a very specific question. We might need more details on that. Yeah.
Okay.
At least to answer on how we have done our cell sorting, we have used the FACS instrument. I don't know if that's something that you have already tried, but if you want to use the FACS instrument, there's also the 10x, but that might be much more high throughput than what you're looking for. So maybe the FACS might be the way to go.
Cool. So next question is less genes detected in automation assay. Is it caused by the small reaction volume with low cDNA input for library prep?
Yeah, so that's a good question. Let me just get this slide real quick. Yeah. So first things is that the manual samples did get a little bit more reads, at 1.3x higher reads. And also this whole study was a preliminary study where the whole thing was sequenced on a MiSeq V2 micro, where you get around 100,000 reads per sample. Usually that's not enough to fully get an idea of correct gene expression in the cells, because you would, for any single cell experiments, you would want to sequence, at least 1 million reads per sample. So if you had gone to that depth and then had a filter that was not Boolean for the genes, I would expect the number of genes per cell to more even out as compared to seeing less genes on the Firefly.
Great, thank you. So next question is, for collaboration purposes, where is SP2 Labtech based?
Sure. So SPT Labtech headquarters is based in the UK. However, we do have 2 sites in the US, in Covina and Concord, both in California. And then our field application scientists and territory managers are sprinkled throughout the rest of the country as well. So you have a pretty significant support team and a sales presence and field application scientist team, both on both coasts as well as in the Midwest.
Cool, thank you. I guess next one is a technical question. So are the Z heights adjustable?
The Z heights for the dispense and the pipetting heads?
The tips, yes. I think the person, yes, the tips.
Yes. I would be happy to dive into that a little bit more with who asked. The software, despite being incredibly easy to use, allows for almost an obscene amount of control over the pipetting for both the air displacement head as well as the dispense head. Happy to run through some examples with whoever that is.
Great. So there's a question, so why is there a difference between the traces for the Firefly pool and the manual pool?
I'll let Ritwik and Cheryl take that one.
Yeah. Let me pull up the slide. Just 1 second. Yeah, so the reason that you see this little flatter area on the manual versus a little peak or a slant here on the Firefly is because the Firefly was better at stopping the tagmentation reaction. So it was very quick and efficient at neutralizing it. So you see more of the library kind of concentrated to like a single area versus the manual, which is you see like a more of a plateau. So that's why you see the difference. It's because the Firefly was much better at neutralizing the tagmentation reaction than my hand.
Great. You also mentioned there's true positive displacement. So what does that mean?
Sure. So the true positive displacement is a technology that SPT first released on the mosquito over 20 years ago, and then is also available on the dispense head on the Firefly. And so what that means for anyone who's ever worked with an air displacement head is that a lot of times when you're utilizing air as the force to move your liquid, it requires a lot of optimization. When you're utilizing true positive displacement, and so do I have my little tool over here? I do. So when you're utilizing true positive displacement, what you have in this consumable that we sell is a plunger that goes inside of this syringe that is in direct contact with your liquid, making it liquid class agnostic. It is very rare that we have to tinker with any of the settings in order to enable accurate aspiration and dispense using the Dragonfly. However, if you do need to tinker, you still have full control of all of the parameters for the dispense head within the Firefly software itself, as well as for the Dragonfly.
Great, thank you. Next question is, what accounts for the gene count difference?
Well, sorry, I think that question was just answered earlier, where I talked about the differences in the gene counts relating to variable sequencing depth. And then, yeah, I think this was just answered earlier.
Cool, thank you. I think the last question we have is, where can I see the Firefly?
I'm glad you asked. So we are currently in Seattle doing some demonstrations for customers. If you want to swing by, just shoot me an email and we can set something up for you. We'll be here today and tomorrow. And then in general, we have quite a few customers who we have really great relationships with who are happy to host people for demos in their own labs, one of those being the Chan Zuckerberg Biohub. We have a couple other sites in the Bay Area as well, a few sites on the East Coast, one being Harvard. And so if you reach out to me or just put an inquiry into the SPT Labtech website, somebody will be able to coordinate with you to set up a demonstration. We also are able to set up personal demonstrations with the instrument in your own lab. That just takes quite a bit more logistics on our end. So a lot of times it's easier for us to just host you somewhere on site with one of our customers.
Great, thank you for that. I believe that's all the questions we have for today. We would like to thank the speakers today again and our sponsor, SPT Labtech. If we didn't have time to get to your question, we'll try to follow up with our experts. As a reminder, please look out for the survey after you log out to provide your feedback. If you miss any parts of this webinar or would like to listen to it again, or an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.